Hello, modern students. Wish all of you are fine. I am Ifatara Jahan, your course teacher, and today our discussion topic is actually reproduction of fungi. And today we're going to learn our second part of reproduction of fungi. In our first part, we already learned about the vegetative reproduction of fungus, and today we'll learn about the spore formation of fungus reproduction with the sexual process of fungus reproduction. So let's get started. So first of all, that is our topics that what is asexual reproduction that we already know. Again, I want to remind that in case of fungus, actually asexual reproduction means that is the reproduction will be processed or happen from one parent. Okay, and asexual reproduction is the most common reproduction in fungus than sexual reproduction. Okay, and the spore formed through asexual reproduction is called the sporulation, and each spore develop into new mycelium, and that is the sporulation of spores. Each of the spores develop into a new mycelium, and these spores are produced as a result of mitosis in the parent cell, and so they are called mitospores. Okay, as in asexual reproduction, the spores are produced from mitosis. It's called the mitospores. And these mitospore actually produce of fungi that are three kinds. One is sporongospores, another zoospores, another is conidiophores. That is three kinds of spores that produced in fungus. So you already know about these three types of spore. In our classification, we already discussed about that. Again, for your memorization, I want to discuss. Okay, that is first of all, that is our sporongospore. So what is sporongospores? It's actually produced inside a sac-like structure. That means the structure will be like that. It is a sac-like structure, and within these structures, all spores will be present. Okay, there you can check that is a sporangium, and whenever the sporangium is break down, the spores come out in the environment. Okay, and these hyphae of sporangium bearing that sporangospores is called the sporangiophores, and they are normally branched, and these sporangiospores can be motile or non-motile. Okay, that means they can be both. They can move, or some spores can move, or some spore cannot move. Okay, and the non-motile spores are actually called the aplanospores, and we mostly found that in mucor and rhizopus. So that is our asexual sporangiospores of fungus reproduction. Then our second one, that is called zoospores. And in case of zoospore, actually we find that kinds of spores especially in aquatic fungus. Okay, and these zoospore are mostly motile. Okay, and it's also called motile biflagellate sporangiospores. Okay, that means you can check there in the figure. In zoospore there are some flagella. Okay, in the bottom portion of that spore, and zoospore are mostly all the spores can motile and they can move. Okay, but they has a lacking, they don't have any cell wall. Okay, and after a swarming period, it secretes a wall and germinates to form a germ tube. Okay, that means after motility, whenever it fixes anywhere, then it secretes a wall and then germinate to form a germ tube, and with that germ tube, it can grow. So that is our sporangiospores and zoospores, and our third one is conidiophores. So what is actually conidiophores? First of all, the conidia are non-motile. Okay, and the mitospores formed in conidiophores extremely as a single separate cell. They develop either directly on the mycelium or on morphologically differentiate hyphae, and it's called the conidiophores. And these conidiophores can be two types. It can be simple or can be branched, or septate or aseptate. And the single conidiophores example of Aspergillus and branch conidiophores, example of penicillium. So these are the three kinds of asexual spore formation of fungus. Sporongospores, that is, can be motile or non-motile, and it's always present in a sac-like fungi, and whenever it's come out, it just sporangium is just broken down and spores have come out in the environment. Then it can grow with a mycelium. In case of zoospores, it's always a motile spore and it's lack of cell wall, and whenever it after swimming period, and whenever it fixed anywhere, then a cell wall is secret and from the germ tube is produced. And in case of conidiophores, it can be motile or these are all are non-motile. It can be branched or single conidiophores. The single conidiophores produce the fungi is called, for example, Aspergillus, and in case of branches is example of penicillin. Then another one is our sexual reproduction. So we already learned about our asexual reproduction of fungus, then the sexual one. So we already know in case of sexual reproduction, okay, there will be two parents always, okay, and the sexual reproduction involves the fusion of two compatible sex cells or gametes of opposite strains, okay, and fungal sex organs are called gametansia, okay, and they may be equal in size or maybe unequal, and there are two names of the male and female. The male gametansia are called antheridia and female ones is called ascogonia. And with that, the fungus may be in sexual reproduction, the fungus may be homothelic or can be heterothelic. In case of homothelic, the gametes come from same mycelium, okay, or in case of heterothelic, that fusing gametes come from different strains or mycelium. And the fungus sexual reproduction actually involves three phases. First of all, it's called plasmogamy, then it's called karyogamy, and last one is meiosis. And these three processes actually occurs in regular sequence at a specific time during the sexual stage of each species. That means these three processes actually simultaneously go one after another in each species of the sexual reproduction of fungus. So here is a figure of that. Before describing the figure, I just want to describe what is plasmogamy, what is karyogamy, and what is meiosis, or what happened in these three processes. After that, I will discuss the figure. So in case of plasmogamy, actually what happened? It's one from the male and other from the female to bring about the nuclei of the two parents close together as a pair. Okay, so in case of plasmogamy, male and female nuclei actually come together and this create a pair, and after that sometimes these two nuclei do not fuse with each other, and sometimes they are fused, and there are some methods of plasmogamy, and each such a cell that is do not fuse is called a dikaryon. And the dikaryotic condition is unique to fungi and may continue for several generations. That means if dikaryon happen, sometimes the fungus will not fuse with each other and they dormant in that conditions for a long time. Okay, and sometimes what happen, that dikaryotic condition is not longer for so much long time, and then these two nuclei divided simultaneously during cell division, and then these are passed on to the daughter hybrid. That means is plasmogamy process. It can be like that. Two parent cell come together and their nuclei can be fused and not fused, both of them. Whenever the nuclei will not fuse each other, this is called a dikaryon. And this dikaryotic condition can be longer for sometimes. So the fungus will be in that position and this dikaryotic position for a long time. But if it gets favorable condition, then this dikaryotic fungus will be go further division, and then they passes its nucleus to the daughter hyphae. Okay, so one of the plasmogamy method is called somatogamy. What happened there? In higher fungi, that's our that we already learned about, that ascomycetes and basidiomycetes. What happened there? In case of this process, just two mycelium will fuse together. Okay, and it will be opposite strain. One is male, another is female. And this post-fertilization changes result in the production of a fruiting body. Okay, which is called, if it is ascomycetes, it is called ascocarp. If it is basidiomycetes, it is called basidiocarp. Then it's go for second process, what is called the karyogamy. What happened there? The fusion of the two nuclei, which takes place in next phase of plasmogamy, is called the karyogamy, and it happened just immediately after plasmogamy in lower fungi, and sometimes in our higher fungi, this karyogamy process is come later, sometime later after plasmogamy. And our third step is meiosis. After karyogamy, these fungal cells go further meiosis process and they produce genetically different spores. Okay, so that is our three process of plasmogamy, karyogamy, and our meiosis. So if you go for now this figure, that what happened at a time in different species. Okay, first of all, in plasmogamy process, what happened? That is a male and that is a female. Okay, whenever these both of these hyphae come together, a fusion happen in nucleus and then this dikaryotic mycelium is produced. Okay, and in some higher fungi, this dikaryotic mycelium can go longer in that position for some so much for 2 or 3 generations, and but if they go the favorable condition, so they prepare that basidiocarp that you already know. Okay, that is the fruiting body. From there, from this mycelium, that fruiting body will be produced. Okay, and they go, and this process is called, then we say that it's our karyogamy process. Okay, in lower fungi, this karyogamy happen just after immediately after plasmogamy, but in some higher fungi, it's just sometime later, or in some just immediately after plasmogamy we can take that karyogamy. Okay, and in this karyogamy process, the nucleus will be deployed. Okay, from there the nucleus will be deployed. When they fuse together, the gamete or mycelium produce this also deployed, and this basidiocarp produce that fruiting body from that mycelium, and then it go to karyogamy till that the your cell or nucleus will be deployed. Okay, after that the deployed nuclei go for meiosis, and in the meiosis process we know our chromosome will be half of its parent cell. Okay, so in meiosis process what happened? The haploid nuclei will be produced, and different kinds of haploid nucleus will be produced that will be genetically differ from each other. Okay, and after that what happened? This from there basically spores will grow, and they will just release in the environment, and whenever they go forward, they get the favorable condition, it will germinate and produce again mycelium, and from there uh, again they go for different kinds of sexual reproduction. Okay, that means after these basidial spores just go for the favorable condition, and then the haploid mycelium will produce, and whenever they if they go the favorable condition for them, the environment, the temperature, the soil, all of is there in favor, then this haploid mycelium can go for again that plasmogamy process, because from there some will be flux, some bacillus spore can be female, some bacillus spore can be male. Okay, Whenever they can produce, they again go for plasmogamy, then again karyogamy, and the meiosis process. And this process will continue. Okay. So that is our sexual reproduction of fungus and asexual reproduction of fungus. And that was for today. We will learn more in our next class. Thank you.